So yeah, thanks for having me, and thanks everyone for their patience. I'll get going. I'm glad to be talking today on phosphorus. I'm glad I'm also following Brian Ott last month, who talks about nitrogen biogeochemistry. So I feel like naturally we have to follow that up with some phosphorus biogeochemistry. So that's what I'll be talking mostly about today. Before coming to NCAR here in Stoneville, I was supposed to talk for the Legacy P project. It's a national USDA-led project. And I'll be talking about that today. And so just talking about a few components of that project. So first of all, there's a lot of people involved here. Pete Kleinman is sort of the boss of the project. Lisa Duranchek is kind of the patron saint of the project. She's at the NRCS. Joshua Mott is also heavily involved, super helpful. And then, of course, then we have all of our site collaborators across, I'll talk about the 7 different sites in a minute, and additional help across the USDA and also some university partners too. So just to give some quick background on why we want to talk about phosphorus, because I'm not sure if everyone in the audience is aware of these things. So the key thing that we're worried about with phosphorus is eutrophication. Eutrophication just means the accelerated nutrient status of an ecosystem. Does anyone know what this picture is that I'm showing?
Is this some sort of algal blue?
It is. So this is from the 1970s. David Schindler, a limnologist out of Canada, he did this, pioneering study on using an entire lake. And what you're seeing is that there's a barrier within this lake. This is like a relatively small, pristine lake somewhere in Canada. On that upper image or upper part of the image, you're seeing basically the control, and on the bottom, the only thing different is the addition of phosphorus. And so there was lots of regulation battles and things about like this. We had phosphates and detergents amongst other phosphorus inputs, and so it took an experiment as drastic as this to really hit the point home of how damaging phosphorus can be to ecosystems. And so it's kind of gone from there. So eutrophication at a very broad, sweeping scale, it degrades aquatic ecosystems, which is in itself pretty bad. It costs us billions. There's estimates in the U.S. That it costs, north of $2 to $3 billion annually as a conservative estimate. It threatens our drinking water supplies and also threatens other, things like fisheries. So this is an image from NASA looking at the extent of dead zones. So dead zones is places where, due to excess nutrient loading, nitrogen and phosphorus primarily, you get hypoxic dead zones. What you'll notice here is down here in the Gulf of Mexico is a major hypoxic zone, and that's there pretty much every year now. When the Mississippi River flow gets a little bit lower and the temperatures start to warm up. And also want to make the point here that this problem is not unique to North America. It's across Europe, Asia, everywhere. And so it's a global problem. Now, why we talk about it in the context of ag? So ag is unfortunately a major contributor to phosphorus pollution. And the reason for that is we need phosphorus for our crop production. Put up here some rough numbers. Typical crop pea uptake, given typical yields and that sort of thing, is 15 to 40 pounds an acre. However, most recommended pea applications are going to be north of that. They're going to be a little bit more. And the reason for that is however much you put out, the plant is really only going to access up to 30% roughly of that pea input. The rest of it can hopefully get from the soil. But yeah, so it's a complicated problem in that we can't exactly deliver precisely what the crop needs in the form of fertilizer. And so traditionally, there's been lots of historical applications where they're building up an excess of phosphorus in the soil. That's kind of like this build and maintain philosophy of pea application. This study I'm showing here from just published this year was looking at soil testing recommendations across North America, or sorry, the United States, and they found that it's all over the place when it comes to the U.S. 19% apply build and maintain, so that's just adding and adding and adding and building up phosphorus in the soil. Meanwhile, roughly a third applies sufficiency approach, so that's just adding precisely what the crop needs and trying to not add much more. And then some states like Mississippi and others are a bit of a hybrid of that. And what's a little bit disappointing is a significant number of states don't even know what the philosophy is when it comes to application. Also, another oddity about Mississippi, we're the only state that uses the Lancaster extraction as like the standard soil testing, soil test P method. No one outside of Mississippi knows what the Lancaster is. I certainly didn't know about it. But most other areas use Malic 3 or Olsen. So I'll be talking a little bit about that. Oh, and I want to point out that Mississippi State Extension has some good resources on their website about pea fertility. I'm definitely not an expert in pea fertility, but looking through what they had, I thought that it was a really good resource. So those of you who are interested. So trying to connect the two. So we have excess phosphorus in our agricultural systems. And then there's this connection to eutrophication and aquatic ecosystems. It's a very complicated problem, one that's been worked on for decades. This image here coming from a paper by Professor Sharpley is just highlighting, a few of the complexities of this and it's kind of like a generalized agricultural landscape. What we can say about phosphorus is that it sticks around. We put it out there and it can stick around for decades or centuries. And so phosphorus that we see in our streams and rivers today is a result of phosphorus that was applied generations past. And that's kind of like a fundamental issue. And that's what gives rise to this notion of legacy P. So this is a map looking at, it's making a lot of assumptions and some, model inputs and outputs of phosphorus inventory across the United States at the Huc-8 scale. But this is a rough indication of what the P balance likely looks like for most of the 20th century. And so you'll see that across the large tracts of the country, we tend to be on the plus side, so excess phosphorus. And for those of you looking at the delta is quite unique in this regard. It's blue. It's actually a bit more negative, at least for this time scale. And I'll try to talk more about that later. And that's just talking about, we're not looking at hydrologic loads of phosphorus in this. This is just inputs, outputs in terms of, dropping systems and things like this. And yeah, so the definition of legacy P that I'll be using in this talk is, it's actually currently a debated topic, but I would clarify it as P in a system that wouldn't have been there in the absence of human activity. So in this presentation, I'll be sharing a few of the insights we have from the Legacy P project so far. There's several different things going on in the project, but I'll just be talking about a couple. So first, I'll be talking about some simulation modeling, an model called Apple. Then I'll be talking about phosphorus chemistry at the national scale across our project. And then I'll be touching on a few kind of specialized topics that we're still developing. And a quick rundown of the project so far. So it's composed of 7 different sites that you're seeing here in this map. And so pretty wide diversity of soil types and chemistries, climates, and ag systems as well. And each one of these sites were picked from the SEEP program, so Conservation Effects Assessment Program. And each of them were kind of noteworthy in some fashion when it came to Legacy P. And so our collaborators across all 7 of these sites have worked diligently with us in terms of sampling and providing lots of information and providing additional studies. And so this has been like a national teamwork effort, and it's great stuff. So I'm excited to be sharing it with you all today. So first, I'll talk a little bit about Apple. So Apple is the annual phosphorus loss estimator. It was developed by Pete Bottas and later on by Carl Bolster and some others. So it's a simulation model for phosphorus losses on an annual time step. And all that it's doing under the hood is using some empirical relationships. So Pete Botta's kind of scrounged literature and put together a number of different relationships for how, if soils have X amount of phosphorus in them, how much can we expect in runoff, how much can we expect in crop uptake, and things like this. And so it's relatively simple in terms of how it approaches this, but it's actually fairly robust, and it's been corroborated for a number of different contexts. So it's quite useful for long-term simulations, annual P budgets, and things like this, and also evaluating the implications of different practices and policies. On the right, I'm highlighting this figure from Jones et al. In 1984. This is kind of actually the core of at least the soil phosphorus chemistry modeling going on. And I'll be showing this again later on in the presentation. So Jones, Andrew Sharpley, and others kind of developed this in the 80s. And this basically cornerstone of phosphorus modeling is still used in phosphorus models today. So it's used in Apple, it's used in SWAT, it's used in a number of different models. So currently Apple exists as a spreadsheet. It looks something like this. You throw in some different inputs. You can put in up to 10 years of different inputs and then simulate for those 10 years. It's pretty simple, but just for basic simulations, it's fairly robust. So we're seeing here total phosphorus loads on a field scale. The lines are what the model's predicting, and then the points are the actual empirical data. And then this is also, so it simulates soil test P as well, using Malick 3 as the common basis. And so again, you're seeing that model predictions are backed up by empirical data. So, and those are the two bits that we're going to be focusing on: is edge of field phosphorus loss, and then also within field changes in soil test P. And so, convinced that the model is working as intended and that it's corroborated. So we think it's a strong basis to start to ask it questions. We can perform different simulations, simulating a variety of different scenarios, management scenarios. So looking at the effects of different fertility regimes, the effects of tillage, and then particularly important to the Legacy P project, the effects of phytomining. And that's just a fancy word for just saying taking out more phosphorus via the crop than we put in. And so these are some simulations from the Lake Champlain site. Joshua Mock ran these simulations, and these, so Lake Champlain is on the border of Vermont, New York. There's a lot of hay production and a bit of dairy and some other things up there. And so this is showing that with phytomining, particularly with corn silage, we can draw down phosphorus, somewhat well, but one thing to kind of keep a note here is that this is a rough time scale, 15, 20 years, and total phosphorus loads on the right, they budge a little bit, but not a whole lot. And so it's just kind of showing the magnitude of the problem, and that it's going to take a long time to address the issue. But at least Apple gives us a ballpark numbers. What can we expect in terms of a time scale? So that's kind of some applications of Apple, but we also wanted to extend Apple to make it more usable. So this is what it looks like currently as a spreadsheet. Rather clunky. I feel like I can say that because Pete Bottas isn't on. It's a little bit clunky, and it's not very flexible, and it just kind of cuts off at 10 years. And so we've been slowly turning this into an R package. And so the point is to make it modular so we can try out new developments, new routines, make it scalable so we can start to run simulations everywhere. And then also ultimately we want to make it interactive. That was actually the original vision of Apple, is that it needs to be interactive. Someone can just simply play with it and see, well, what happens if my soil test P is higher? What happens if it rains really hard? What happens if I change my management? So that's kind of what we were working on this part here. So I'm showing here, example R script. just showing for those of you who speak R, just trying to show that it's relatively simple what we're developing here, and that you fill out the data template, it reads everything in, and then you run basically one function down here, and it runs Apple, and away you go. And then we're going to be adding a number of different utilities, plotting functions, etc., and so that users can quickly start to look at the dynamics of phosphorus with Apple. And then this is the interactive bit. So I'm still learning a lot here, but the goal is to put this into a Shiny app. So I don't know if any of you are familiar with RStudio Shiny, but this is kind of what that looks like. So I've got a very rough demo put here, and so we can play with a few buttons.
You make this?
Yeah.
Cool.
So you can change what the initial soil test P in the simulation is, and start to look at what that changes in terms of the outputs. So I'm going through here, changing a few different bits. And so this is, just a demo version, but the intent is that we'll have it where users can upload their own data with a template, or they can use some, ready cooked examples, and then they can start to play with even more buttons and widgets here than I have here. I've just got initial soil test P as kind of a demonstration here. And so we're looking here on the right, inputs and outputs. You're seeing if I put the soil test P up to 500, something really excessive, you can see how that changes with the outputs. And so we'll have more there to come. So the second bit I wanted to talk about from Legacy P project is this national study of soils and sediments. So across the 7 different sites, all the teams collected a total of 624 soils and sediments. And these are from various different fields and catchments. A huge, and so, roughly 80, 90 samples typically across each of the sites. And all the analyses were conducted at a centralized lab in Pennsylvania. And so Kyle Elkin is the one who's most responsible there. But it was a huge analytical effort. It took a lot of troubleshooting to get everything to work out, and also to get everything consistent across all these different samples. Things change dramatically across these different 7 sites. Snake River in the west there really throws us for a loop because it's more of an arid system and has more alkaline soil chemistry. So everything about that was weird, but we were able to kind of manage. So I'll be showing just a few of the highlights from this data set. So
you can hide it if you click on more. Yeah.
Or this one.
That one. Yeah.
Okay. So this is just a quick snapshot of some of the general characteristics of these soils and sediments. So when you have a huge range in pH, roughly 5 to 8.5 or so, an enormous range in clay content, organic carbon from very small up to pushing 100 grams per kilogram. Total P up to I think the highest was 16, 17, maybe even 1800 milligrams per kilogram. I've seen higher, and I was expecting higher for this project, but even still, that's quite a bit. And then Malic 3P, I don't know if any of you have an affinity for what Malic 3P typically looks like, but it varies enormously state to state what recommendations are. Agronomic optimums are usually 40, 50 milligram per kilogram. Some of the samples from Pennsylvania and Maryland are north of 400, 500. It's insane. So we definitely got a huge magnitude of phosphorus in some of these things. So as I said, I'm bringing back that image from that Jones et al. Model. So part of what we used in the Legacy P project, we wanted to be informed by models, but we also wanted to use this data to inform models, and so to kind of have this feedback. And so SWOT is one that we think about a lot. And so kind of the kernel of the P routine in SWOT is this, simple 3-pool model. So these 3 different pools of labile, active, and stable inorganic P. nothing much has really changed there since the 80s. And so we really wanted to drill into this. And I'll mention here that so labile P is the mass of phosphorus that can readily exchange with solution, readily meaning on the timescale of some hours or so. And that's through desorption, so no further chemical mechanism there. The active and the stable pools are more so hypothetical, based off of what we kind of like a very, useful simplification, let's say, of P chemistry in soils. And so what we decided to focus on in this study in particular is labile P. And I'll be explaining why. But to build up to this, I need to give a very quick rundown of P chemistry in a nutshell. So those of you who have seen the absorption isotherm, on the y-axis we have net absorption. So here I'm just showing positive. So this is the forward reaction of phosphorus absorbing onto a soil or sediment, and that's a mass per mass soil or sediment. And then on the x-axis you have an equilibrium concentration, so mass per volume. So as we can have different points along this curve of where, let's say, a soil is located. And the slope at that curve also tells us sort of the P buffer intensity at this point. And of course, each soil has its own unique curve, and depending on the status or its history of phosphorus application, it can be at various points along this curve. So labile P is the pool of phosphorus that could release back into solution on the timescale of hours. EPC is the equilibrium phosphate concentration at net zero absorption. So that's the x-axis coordinate where no net absorption is occurring. And the P buffer capacity is the slope at the EPC. So how strongly does a soil sediment buffer phosphorus in solution? So labile P, we're focusing on that quite a bit. If we express labile P in the soils in the project as a stock, that's what this top panel is showing. So just assuming top 1 centimeter of soil, a bulk density of 1.2 grams per centimeter cube, this is what those stocks look like. So typically around 10 kilograms per hectare or more. Typical edge of field phosphorus loads and also small catchment loads are what the middle and the bottom plots are showing. So this is from the managed database created by Darren Harmel. It's a collection of edge of field and small catchment phosphorus loads on an annual basis. And so in the middle, you're seeing subsurface loads through tiles and another subsurface flow paths. And on the bottom, you're seeing surface runoff. And so you're seeing dissolved and total P there. And so, and I want to point out that all these scales are on the log scale. And so the point I want to make here is that there's enough labile P in just the top 1 centimeter of these soils to sustain these kinds of phosphorus loads for a long, long time. So that's kind of the tip of the iceberg that we're seeing. That kind of scares us. So when it comes to predicting labile P, very often in literature, we use soil test P as the basis. And the reason for that is because soil test P is readily available. It's what's commonly measured. Labile P, as we measure it here, is a little bit intensive. It was a 24-hour desorption. And you have to have it in the presence of a phosphorus absorption sink. So it gets a little bit complicated. And so it's not typically measured, but we measured it, 600 plus times. What this plot is suggesting to us here is that there is no one general relationship between Malic 3P and labile P. Labile P is the important bit. That's what determines what can come off or runoff. And typically we say if Malic 3P is X, then this is what you can expect to run off. And that's like a blanket application no matter the site and whatnot. So for example, Apple is guilty of this. It initializes the same. But what we're seeing here is that blue and the brown lines here, these are the sites in Pennsylvania and Maryland. They have a relationship that comes way out here, a little bit flat compared to the others. Whereas over here on the left, you got Beasley Lake. That's our site in the Delta, like just 20 minutes drive from here. It's this kind of light purple color. It has a really steep relationship, as does quite a few of the other sites, particularly sites where they have a rich amount of clay content in their soils. So this is one of those, relatively simple but kind of profound results from this study, and that it seems like no one has really picked up on this in the literature, that soil test P is actually kind of a terrible predictor, and there's very good reasons for why that is. And so terrible predictor as in it doesn't generalize well. So if we wanted to predict, what happens in the middle of Ohio or something, or southern Missouri or whatever, a place that hasn't been sampled like this, it would be a little bit tougher to say accurately what that relationship would look like. Another thing that we included in this project is EPC. So EPC is where the net absorption is zero. So neither is the concentration in solution where neither absorption or desorption is occurring. And it varies dramatically depending on the P chemistry of the sample, but also depending on phosphorus history of the sample too. So samples that have been exposed to more and more phosphorus, they typically have higher EPC. Normally we measure EPC with several different centrifuge tubes and equilibrations. You have to kind of build this relationship and plot a line and then find that x-intercept. Kyle Elkin told me, oh, no, I'll give you like 3 tubes per sample because I'm not doing that many. And so we had to kind of do some statistical jujitsu to make this work across the project, but we have it. So we have 600 plus measures of EPC. And as far as I'm aware, that's the largest data set of EPC in current knowledge. And so EPC and label P have a very strong relationship, we found. So on the left is the raw data you're seeing, so just label P versus EPC. And then broken down according to a generalized additive model on the right, we see that we have this very strong concave curve as a function of EPC, and then these lower 3 panels for clay content, Bash-Williams index, and iron to P ratio, which I can talk about later. But those are kind of proxies for the buffer capacity of the soil sediments. And so what this tells us is, at a given intensity, more phosphorus can release in the solution given a greater buffer capacity. And this is kind of backed up by really old classic theory going back to the 60s. But it's really exciting to see it at this scale and something that generalizes across soils, sediments, swampy environments, up hill slopes, and then across a huge range of different chemistries. And so something that generalizes this well across the project is really exciting. And going further from this, so label P isn't something that's going to be typically measured, and neither is EPC. But we're trying to think through what's something that can be typically measured that can help us approximate these things. So using some of that same, classic soil peat industry stuff, think about quantity, intensity, capacity relationships. On the x-axis, we're looking at Olsen P, so a pretty common soil test P measurement, and then the Bash-Williams index, which is a simple metric for the P buffer. It sounds fancy, but it's actually extremely simple to measure. It's just not commonly measured. So a simple ratio of these two actually gets us much, much closer to a generalizable relationship across the 7 sites, with Indiana at the Western Lake Erie site being a bit of an exception there. But what this is telling us is that there could be ways to simply approximate this, and that's more exciting. That'll be a great step towards building better phosphorus models. Okay, so last bit I was going to share in my presentation is kind of more to come. There's a number of different little offshoots in this project, and there's also a huge modeling component that I'm not going to talk about, but Mike White, Amy Collick, and some others who are the SWAT wizards, they're running SWAT at unprecedented scales, and they're really drilling into some of the phosphorus routines, and so they'll have a whole lot more on that to come out of this project. But since I care more about, or I'm more involved with the P chemistry work, I'll show a little bit of that. So we also have some heat absorption kinetics stuff coming out of this. We can only do this on a subset of the samples across our 7 sites because it's rather intensive. But we think here that there's a lot to really dig into. And so we're seeing a huge variety in terms of how fast and how much phosphorus can release into solution. And this could be something that could be implemented in models to describe like, well, for a 1 hour storm event of a given intensity that generates X amount of runoff, this is the time that the soil is given to exchange phosphorus into solution, and this is how much we could possibly predict given the characteristics of that soil. So using this kind of data, we might be able to further improve models in this regard. Another exciting development, so Kyle Elkin, the research chemist out of Pennsylvania, through this project, we got him in his lab a laser ablation ICP mass spec setup, so LA-ICP-MS. And so this is a novel method that hasn't really been used much to look at phosphorus in soils and sediments. And so all it is it's an ICP-MS where instead of injecting a solution to speciate phosphorus and a number of different cations, it's using a high intensity laser to ablate a soil sample, turns it into a plasma, and then sucks it into the ICP and it can spit out numbers. And what the magical part of this is it can do this extremely fast and in a spatial dimension. So we can get these 2-dimensional images across soil sample, a number of different elements, including phosphorus. And so we think that this will be kind of an advancement in terms of looking at total phosphorus in soil sediments, particularly because we'll be able to get a more clear picture of the speciation. So, do we see a pocket of phosphorus that's associated with calcium or iron, aluminum, etc.? So this is another shot of what that kind of looks like. You get a soil pellet packed in, and just looking at a very small area, we can get a very high resolution image. I forget what. Oh yeah, Kyle said it was so 300 square microns at a 2-micron resolution. So a really fine resolution looking at pockets of phosphorus and calcium in this image. But that there's, this is a brand new methodology and there's a whole lot more troubleshooting here. But it's something that we're really excited about in the project. And I believe that's all I had today. So yeah, thank you.
If anyone has any questions online or in the room?
I have one question. You mentioned like the phosphorus loss. Is the phosphorus loss is like I'm confused about the jargon. Is the quantity of phosphorus or you are measuring the loss of phosphorus?
So is the question, what is phosphorus loss?
Yeah, it's phosphorus accumulated or phosphorus loss from the field.
So I'm talking about it generally in terms of a load leaving the field or a system. So edge of field phosphorus loss would be kilograms of phosphorus loss per hectare per time step. But that's what that is. But there's also subsurface phosphorus losses too. So leaching losses through the profile.
So in that case, what is the main factor that transport of phosphorus weight? Like what is the major component input? Like a runoff or like primary to drive some more transport towards the downstream of the river, the phosphorus.
Did I understand the question to be what is it that drives phosphorus loss?
And what drives the phosphorus loss?
What's the form of it?
What's the
Because you asked if it was in the runoff or if it was dry matter or if it was.
Right. So
phosphorus.
environmentally dry it's what's carried by runoff. So it could be suspended particles, so soil erosion itself that's carrying this particulate P with it. It can also be a dissolved P, so phosphorus that desorbs from a soil into solution, and then that's carried by water. So in a nutshell, that's what phosphorus loss is. Hydrologically, yeah. there's also phosphorus removal through, crop uptake and things like this too.
I think you have an online question.
How is the model affected by the form of P applied and also when it is combined with other fertilizers such as nitrogen and potassium? Great question. So for apple, it considers inorganic and organic fertilizers separately. So inorganic, it just runs some simple routines about how much inorganic phosphorus is there. And most of that's going to be pretty much immediately available as soon as it gets solubilized. But then there's of course some fixation and such that happens there. With organic phosphorus application, so pretty much just manures, that gets a little bit more complicated. And so right now Apple simulates a number of different poultry and cattle manures, to my memory. And there's some organic P mineralization rates built in. And so depending on the time of application, like what season, how much, and et cetera, it simulates what typical organic P mineralization is going to look like. And there's also a bit of a dynamic pool built up. So if it's not mineralized in that current season, it might be mineralized in the later season. So in a nutshell, that's how some of those different fertilizers are handled. In combination with other fertilizers such as nitrogen and potassium, I don't think Apple really simulates much there. I would say, if anything, there would just be the indirect effects on fertility. So if you have more nitrogen and potassium and it promotes a greater crop yield, that could be removing more phosphorus from the system. So hope that addressed the question.
Any other questions? All right, thank you for coming.
Thank you.